Hello, and welcome to this short tutorial about the main functionalities of IDdSV. Concerted efforts have been made in sequencing, analyzing, and sharing SARS-CoV-2 genomes all around the world to control the spread of the virus. The International Database of SARS-CoV-2 Variations IDdSV is the result of ongoing efforts in curating, analyzing, and sharing comprehensive interpretation of SARS-CoV-2's genetic variations. Through a user-friendly interactive data visualizations, we aim to provide a robust surveillance tool to the scientific and public health communities. The database is accessible through the following link and enable to its users browsing informations about over 60,000 variations. The graphical user interface was designed to provide an easy access to all functionalities of the database. Users can access all the implemented options from the home page. Then select a variation to get more information about it. IDdSV provides to its users three main ways to navigate through the identified variations. Users can access information about a variation by clicking on the View from the Variations table on the home page, by the position of the variation in the genome, or the position of the variation in a specific gene. Now, let's look at a real-world example. The 28,281 A-T variation is a mutation located in the gene coding for the nucleocapsid protein. Several scientific papers link this mutation with a variation of the B117 lineage. Yet, no further information about it are available. Neither its origin is known. Let's see how to retrieve information about this variation from IDdSV. Users can first check the variations table. This table gives summary about all variations available on the database. The table is composed of 6 columns and a clickable link for more information about the selected mutation. If the desired mutation is not visible on the first page, the users can navigate up to 500 pages of this table. In our case, the T881AT variation is on the second page. From the table we can see that variation 28,281A-T is a nucleic change of an adenine to thymine, and it is located in the third position of the nucleocapsid gene and encode for an aspartic acid change to valent amino acid. To get more information about this variation, users can click on the View button. If a variation is not visible from the main table, users can use the search tools to launch search queries. The Search by Nucleotide Position box allows the user to browse variations simply by motioning the reference, alteration, and position of the variation in the genome. In the case of the 28,281 A-T variation, the reference is A, the alteration is T, and the position is 28,281. Similarly, users can also browse the database using the position of amino acid variations in a specific gene. Again, in the case of the 28,281 A-T variation, the reference amino acid is D, the altered amino acid is V, the gene is N, and the position is 3. Once a variation is selected, the platform retrieves for users 5 types of information. The first section includes information about the DNA mutation and its position in the genome. The second section provides information about the functional annotation of the variation. This includes the amino acid changes, the associated gene, the position in the gene, the type of mutation, and the predicted impact of the mutation. Here the database shows that the D to V variation in the third position of the N gene is a missense structural mutation with an impact estimate to be moderate. These data can be downloaded for further analysis by clicking on the Download button. The third section gives information about the first no appearance of the selected variation. The date, location, and the ID of the strain related to the first appearance are shown in the left panel, while the right panel shows the location in an interactive map. If users want more information, they can use the ID to retrieve more information about the sequence from the GISED database. Here, for example, the database shows that the selected mutation appeared in February 7, 2020 in Spain. Finally, the last section gives information about the associated lineage with this variation and its frequency among the analyzed samples. More information about the frequency of variations is available in the Frequent Variations pages. Here, the top 20 frequent variations are displayed. This table is updated on a monthly basis to keep up with the continuous evolution of SARS-CoV-2. Furthermore, the frequency of variations can be visualized geographically from the Geographic Location page, where frequencies are calculated for each continent individually. For more information about the data analysis pipeline, samples and statistics, you can visit the web page and the manuscript.